Okay, so again, I'm Eric Admin with Sno-King Watershed Council, and welcome to Stream Biomonitoring. So what is stream biomonitoring? And also let me know if my Zoom bar gets in the way of you seeing anything. I've had that happening on some of these classes before. So what is stream biomonitoring? It is essentially a way that you can look at the health of a stream, the ecological health of a stream, by looking at the creatures that live in it. And so we're talking about something that we refer to as benthic macroinvertebrates, which sounds complicated, but essentially benthic means it lives in the bottom of a stream, typically bottom of a body of water. And macroinvertebrates are animals that don't have backbones, but they're naked, they're visible to the naked eye. These include a number of different types of animals. So crustaceans, so things like crayfish, insects, like mayflies, for instance, mollusks, so clams and worms are some of the things that we look at. And they're good indicators of stream health. There are other methods of assessing the health of the biological community of the benthic macroinvertebrates, and one which is used by scientific agencies and government agencies is called Bibi. And I should have written out what the acronym is, but it stands for Benthic Index of Biotic Integrity. And this is a different method where you use a particular type of a net, and you can see one of our volunteers, Leora, using that particular type of net there. And it collects, what you do is you stir up a measured amount of the bottom for a prescribed period of time. You collect those organisms in net, and you do that 8 times to represent 8 square feet, or 3 times to represent 3 square feet. You preserve those samples in alcohol. You send them to a taxonomic lab. A scientist at a taxonomic lab evaluates those and then gives you a score. And so this is something that agencies use to quantitatively assess and compare sites. So if you go onto a website called Puget Sound Benthos, you can look that up, that data that's already been collected in different projects, and there's a really useful map. So what's great about this technique is that it gives a relatively standard comparison from site to site. So you can go, okay, well, this one's relatively good and this one's not as great. The downside to it is that it's expensive because it costs about $300 a sample to process. And from like a volunteer standpoint, it's not quite as interactive. You don't actually get to really see the organisms you're collecting yourself very much. You just take a collecting container fill it up with alcohol and ship it off. Whereas the stream biomonitoring, we're actually going to look at the creatures, identify them, sort them, and then release them back. So, but I just wanted to let you know that there are different methods of assessing what lives in the bottom of a stream. So there's some reasons why we use these creatures. And one of them is that they're pretty easy to collect. Doesn't take a lot of work. There tends to be quite a bit of them in the bottom of the stream. In fact, you'll be amazed when we do our collection how many we come up with. They tend to stay in one little area of a stream, so they don't really range very far up and downstream. So they give you a good idea of what's happening in that particular reach of the stream. And they also live for a long time, sometimes more than a year. So they may spend maybe 2 weeks or just a short period of their life out as, say, a flying insect, but they've spent about a year living in the stream. So again, because they're long-lived, that gives you sort of a sense of what's been happening in that stream during that whole time period. So that's another reason why they're a good indicator. And the key thing that's about them that makes them useful for our purposes is that they have varying levels of sensitivity to pollution. So some of the types of creatures you find are quite tolerant of pollution, where some of them are quite intolerant of pollution. So if you find mostly those ones that are tolerant of pollution, that probably tells you you have a water pollution situation. Where if you find mostly ones that are pollution intolerant, or you find a good mix with a lot of pollution intolerant, that might tell you you have less pollution and more of a clean stream situation. This is an illustration from the Keeper. Yeah.
When you're going through this, can you identify the various macroinvertebrates on the slides? Thank you.
Sure. Yeah. So for instance, this is a case-forming caddisfly right here that we're looking at. So they come, you're going to classify them into different large groups. We're not going to classify them down to the very detailed level. So we're essentially going to be looking at like the order and family level. But overall, this is the classification of the benthic macroinvertebrates that we're going to be looking at. So you've got some worm type creatures. You've got segmented animals like crayfish and insects. You've got clams and snails. Those are kind of the different types of creatures that we're going to find. And primarily we're going to find, well, we're going to find all those different creatures. So one thing that will help you in identifying the different creatures is to understand the anatomy, and we'll use guides and pictures to look at the creatures to help identify them. But just in terms of talking about the different anatomic parts, we describe some of the different parts of the body, like the head, the thorax is kind of the below the head portion. On an insect, you may see wings developing. You may also see gills, and you'll see tails. And for instance, on the mayfly, the number of tails is a key indicator. If you see the 3 tails, that tells you that it's a mayfly. Now, a caddisfly looks a little bit different. It's an insect as well, but you can see the shape is different. You might think that it has lots and lots of legs, but those are actually gills on the underside of that body. And then a midge has just doesn't even really have legs that you can see very well. But you'll also see some characteristic movements of these creatures that'll help you identify what they are. Depending on the creature, insects in particular, they go through different life cycles. So some of them go through what's called incomplete metamorphosis, and some go through complete metamorphosis. And incomplete essentially means they go through more stages. Fewer stages. So incomplete basically goes from egg to larva to adult. Complete goes through egg to larva to pupa to adult. And we're mostly going to be finding things in the larval stage. They're also categorized in terms of different, what are called feeding groups. And these insects and other creatures are a key part of the food chain. So a lot of these serve as food for fish and other macroinvertebrates. So what they do, they typically eat things like leaf particles, fungi, algae, and so depending on what they do and how they eat, they're classified into these different functional feeding groups. And that also varies on where you're going to find them within the stream continuum. So we talked about that stream continuum before, from kind of the headwaters down to the mouth of the stream. So for instance, the shredder group, we're going to find more in the headwaters, where there's a lot of leaves falling right into the creek. Collector gatherers are going to be kind of more a middle and low reach. Scrapers are things that feed off algae and things like that attached to rocks. They're going to be a little bit more where there's some more sunlight hitting the rocks. And then predators are going to be throughout. So depending on what type of a place we're collecting, we may tend to find more of one group of creatures than another group of creatures. So if we're collecting more in a headwaters area, we may get more of the shredder group. And if we're collecting more downstream, we may get more of the scraper group.
Hey, Eric, I have a question.
Sure.
When you said that we are likely to find more macroinvertebrates in the larval stage, is that just because they die as they get older?
It's because they spend most of their life in that stage.
Okay.
Yeah. Okay, so I mentioned that there's different, we talked about the stream continuum, but then even within a given reach of a stream, there's different habitats. So there's what's called a pool, which is sort of a flat section of water where there's not a lot of water moving. And then where it's flowing out from that pool over some rocks, it's well aerated, is what we call a riffle. And then a run is kind of a long straight stretch where it's just flowing without a lot of turbulence. And typically we'll find the highest collection of macroinvertebrates in a riffle at the end of a pool. So that's typically where we're going to focus for our collecting efforts. And it's also, we want essentially to have our best chance of collection. We want to collect in a stream that has beyond a certain minimum level of flow. It's about anywhere from 6 inches to 2 feet deep. And we have a good riffle at the end of a pool. That's going to be a good environment for us to collect in. I mentioned that they're more or less tolerant of pollution. And so that's why they're indicators of how polluted a water body is. And for our purposes, we're going to put them into 3 different groups: group 1, group 2, group 3, from the least tolerant to more tolerant of pollution. So group 1 would be these different organisms here. So a stonefly, riffle beetle, mayflies, caddisflies, and water penny. And caddisflies are pretty interesting. They come in a pretty wide variety of forms. So you've got from that top left caddisfly, I think is probably either a free-living or a net spinning caddisfly compared with the case forming caddisflies, but they're all different forms of caddisflies. And then mayflies are pretty easy to distinguish because they typically have 3 tails. Stoneflies typically have 2 tails, but when we do the identification in the field, we're going to be using photographic guides, diagrams, and we also have a key that you can use to help identify them. So that's group 1, common creatures. Common ones in group 2, there's a different form of a caddisfly that looks like that. There's a helgramite, that one that's called an amphipod, we also refer to as a scud, and it looks almost like a little shrimp when it's swimming around. The black fly is pretty easy to identify because it sticks to the bottom, or it sticks to whatever it's on, and it waves back and forth. It almost looks like one of those things that you see on the side of the road. They put a fan in the bottom and it blows up and waves back and forth. That's kind of what they look like when you're looking at them in the water. And then you can see some of these other creatures that we find in group 2. So again, depending on if we're finding more of group 1 or group 2, that's going to affect our index value. And then group 3, most pollution tolerant, includes things like midges. So those clear ones on the top left and the red ones below, those are midges. And those are pretty easy to identify. They look kind of like a segmented worm almost, and they move. Their shape's kind of like a letter C, and they just constantly sort of twitch like a closing up the letter C. So those are pretty common. We'll find those. Aquatic worms. Snails, will find that depending on which way the snail opens is either a pollution tolerant or not a pollution tolerant snail. And we've got diagrams for that as well. And then leeches is another thing that's pretty pollution tolerant. So let's talk about what you need for.
Can I jump in with a question?
Sure.
I'm not going to be able to get it down to this fine of detail, but is it known whether like any of these are not like native macroinvertebrates, or like is it even researched enough to understand that?
That's a great question. There are some macroinvertebrates that are non-native that are problems, like New Zealand mud snails, for instance. But I don't know about all these other ones. They seem to actually be widespread worldwide. So if you look at these, you'll find a lot of these same same sets of macroinvertebrates in streams all around the world? Good question. Okay, so let's talk about the basic equipment that you need for biomonitoring. So you're going to need some nets, some buckets and some tubs, some things for picking up and collecting and moving your macroinvertebrates, some guides so you can figure out what you're looking at, some magnifying lenses, what we use are sorting sheets to help us put them into different categories so we can identify how many we have of each kind. Some tables are helpful, and you want to be dressed for the weather. And then ultimately, we're going to record our data on a biomonitoring data form. So there's different kinds of nets that you can use. Two of the nets that we use commonly for sampling, depending on the size of the stream or the habitat that we're sampling, is either a D net or what they call a kick net or a seine. And so the D net is a smaller net. The mouth of the net is shaped like a letter D, and you can use that for, you place that on the bottom of the stream and you could stir up the bottom in front of it, or you could use a larger net, which allows you to stir up a little bit larger area in front of the net and then collect your sample. So those are a couple nets that we use. We'll use tubs. We may use buckets. So what we do typically is collect our sample in the net, and then we'll put it into these tubs, and then from there we'll sort them out and put them into sorting sheets, into little petri dishes put on a sorting sheet. So these are some other pieces of equipment we're going to use. We use a sorting sheet just to help us group what we're finding. And so we'll put little dishes in each of these spots on the sorting sheet and set this on a table. So as we find different creatures, we'll put them into the different categories. And then we're going to record that ultimately when we're done, we're going to record how many we found of the various different taxa on our biomonitoring data form. So let's talk about the process. So again, I mentioned that we sample in a riffle, and we want to approach from downstream. So you're not kicking up the bottom where you're going to be collecting your sample. And the best is a riffle at the bottom edge of a pool. What I found is that for sampling in the riffle itself, you can see that's a good sized net that is placed across a stream. And then what we do is you weigh the bottom down with a couple of rocks so that no water flows under it. And in front of it, we're going to pick up larger rocks, rub off anything that's attached to the rocks. Because what we find is these creatures are they're attached to rocks, they're attached to leaves, they're attached to pieces of wood, and they're also burrowed down into the bottom. So we let the water flow through this net, and immediately upstream of the net, we're going to essentially disturb the bottom a little bit by picking up rocks, rubbing off what's on the rocks, and then ultimately kind of digging up the bottom a little bit in front of the net, essentially trying to come up with about a 3 square foot area in front of the net. So we're going to use that to sample in the riffle. And if there's other habitats around, for instance, there's overhanging leaves, grass, other things like that, we might use our D net to help shake off some creatures that might be on that and collect those creatures as well. So that's kind of step one, is collecting our sample with one of these nets. And as I mentioned, there's different microhabitats. So it could be the bottom at the riffle, it could be overhanging roots or leaves, could be pieces of wood. Dead leaves are another great place that you can find things, because these creatures, a lot of them that's their food source is leaf debris. And so you'll find a bunch of them on dead leaves. They stick to rocks. They might be on the edges. So we're going to try to collect a little bit from some different habitats when we're out on the site. And then in the bottom, of course. So once we've collected our sample on a net, then we're going to transfer that sample of creatures into a tub or a bucket. Ideally, a tub is more useful. And if we have a large sample, we might put it into multiple tubs, or if we have multiple people working on it, we might put it into multiple tubs, just so that you can get your sample spread out. And essentially you want to have a little bit more water than what you need just to cover the amount of stuff that you've collected in the tub. And then what you'll do is you just kind of let it sit still for a bit, and you'll start watching for things that are moving. And then as you find things that are moving, you're going to collect them using either a spoon or a pipette to kind of suck them up, and we'll put them into our sorting sheet. So in some cases, there may be some remaining on the net. There may be a little bit of net picking. There's going to be identifying what's in the tub and pulling that out. And then we're going to put them, in this example, they're putting them into ice cube trays, which is another common way that people sort them. We typically use this thing called a sorting sheet. And another thing that will help is the magnifying glass, so you can look at what is it that you're trying to, what is it exactly that you're looking at?
And these tubs and containers all seem to be white.
Yeah, that, that's helpful, but probably not necessary. That's just what we've used. And the other thing I should say that I didn't list here that we use is I have a variety of different guides. So it helps. The most helpful thing is to have pictures with names, so that you go, okay, that looks just like this. Okay, I know it's a mayfly, or I know it's a such and such. So our guides are, most of the things that we find around our guides. Once in a while, there's something we find that's not in our guides. So I have some other reference materials I bring along. But there's also a thing called a dichotomous key, where you go, okay, does it have segmented legs? No. Okay, does it have a body like a worm? Yes, etc. That can help you sort things out. But the quickest seems to just be using the photographic guides.
Depending on the time of year in the life cycle, we're getting into fall. So would we see different stages than what we would have seen in the summertime?
That's a good question. And it is possible that we would see different stages. Up until about early October is a pretty good time for collecting in this region. The one thing that can affect your results is if you have a lot of heavy rain, it can kind of flush out creatures or make them hide more. And so that can change what you're finding. So your results, maybe you have to factor into the fact what the weather is and what the stream flow is. So like we wouldn't, if we went out to do this and we found ourselves in a high stream flow situation, we would probably have to come back and do our collection at another time. But up through early fall, August, September into early October, around this area is a good time for collecting. Okay, one of the things that we're going to do, and there's different ways that you can do this habitat assessment, but we're going to make some observations at a site. So the idea would be you're doing this once a year at a site, and you want to document how's that site changing over time. So there's some ways that you can assess that, and you're going to look at things like, what's the tree canopy cover? Is it mostly open? Is it mostly shaded? What's growing around the stream? Is it covered by, are there overhanging trees, or is it just bare dirt right up to the edge? What's going on in the surrounding environment? And so you can record this on your site, so that next time you go do that site, you can compare and go, well, last time it was all trees, now all the trees are gone. So that might help you put what you're finding in context. So that's part of the process. And then you're going to record the results, and we'll talk about how you calculate the biotic index. So on this form, there are different, there's different spots on this form for you to record the different types of things that you find. And as I mentioned, this particular index method here groups them into 3. There are some other indices that use 4, But essentially the idea is to come up with a weighted number based on how many different types of things you find within each of these categories. So you're going to count how many individuals are in each taxa, but we're primarily concerned with how many taxa we find in each group. So for instance, on this form, group 1 is caddisfly, mayfly, riffle beetle, a particular type of snail, stoneflies. So if we found all those things, 1, 2, 3, 4, 5, 6, we would multiply that by 3 and get an index value of 18. So then if we looked at group 2 taxa, let's say we found, essentially the more you find, the better. But let's say that we didn't find any group 1, we didn't find any group 2, and we just found one of each in group 4. You can see that the weighting factor at the bottom of the group 3 taxa is only 1. So our number of taxa would only be 4. And then when we added those all up, after multiplying them out, we'd have a stream quality of poor. Or if we found a lot of group 1, some group 2, and group 3, we might have a higher number. And so that's how that weighting process will work. So again, we count up the number that we had in each taxa, and we'll note, just for future comparison, whether they were rare, common, or abundant. And that's where you do that on the form. We're going to enter that code in the spot for the taxa, and then it will leave it blank if we didn't find one. And as we go along here, feel free to ask questions or stop me if it doesn't make sense. So then we're going to record the number of taxa per each group, multiply it by the factor to come up with an index value. And again, it's weighted. So the more of the pollution intolerant group 1s that you find, the higher of a number you're going to get. Where if you mostly find pollution tolerant, you're not going to end up with as high of a number. Then you add up your index values and your taxa, and the stream quality assessment is going to be based on your cumulative index value. And that is pretty much the process. So I'm going to stop right there, and I'm going to stop the recording, and then I will take any additional questions.